The next set of topics that we're going to be delving into in the course are all related to reproduction and genetics and the passing on of traits and characteristics. And we're going to begin this discussion at the cellular level by looking at different ways that different types of cells can divide and produce new cells. Since the evolution of cellular life some 4 billion years or so ago, continuity of life, the production of new cells to replace old cells, depends on cell division. And depending on what type of organism you're talking about, that cell division is going to occur in different ways. So simple cells can divide and produce new cells by a relatively simple cellular mechanism, while cells that have more complex structure are going to have to rely on a more complex mode of reproduction. That makes sense. So what we're going to do in this discussion is look at the three main types of cell division, and we're going to focus most of our time on the types of cell division that are occurring in eukaryotic organisms rather than prokaryotic organisms. So the three types of cell division mechanisms that we're going to be talking about are binary fission, mitosis, and meiosis. Binary fission is a simple type of cell division that's used in prokaryotic organisms to produce new daughter cells. It's a simple form of cell division, and it can be used because these organisms are structurally and genetically simple. We've talked before in the course about the fact that prokaryotic organisms lack internal compartmentalization, so they don't have internal organelles. They have one common cytoplasm, and all of their biomolecules are present within that same cytoplasm. In addition, we've talked about the fact that their DNA is almost always found in the form of one circular DNA molecule, and maybe a few extra chromosomal plasmids that are not essential for life. So genetically speaking, they're very simple. Binary fission is the means by which prokaryotic organisms produce new organisms or propagate the species. The daughter cells are going to be identical clones of the parent cell, so this is a form of asexual reproduction. When will bacteria or archaea cells undergo binary fission? Primarily, they're limited by nutrient availability in the environment. As long as nutrients are readily available and wastes have not accumulated to toxic levels, these cells are going to be dividing. For example, if you have E. coli growing in culture in the laboratory, and you provide them with plenty of nutrient broth, they will undergo binary fission and divide every 20 minutes. So essentially, the population doubles every 20 minutes. Let's take a quick look at how binary fission occurs. Here's a prokaryotic cell, let's say an E. coli, that has already replicated its DNA. So just to make clear what we're looking at here, we started with a cell that had one DNA molecule and it's been copied. And now the organism will need to keep track of and control the separation of their copied DNA to make sure one copy goes to each daughter cell. To do that, they use this interesting little trick. They attach their DNA to the inside of the cellular membrane. The DNA gets copied, and what we now have is two circular DNA molecules that are sort of intertwined and attached right next to one another at this inner surface of the cell membrane. The cell then starts to grow and elongate by inserting new cell membrane and cell wall here in the region between the two attachment points of the DNA. This essentially pushes the copied DNA molecules away from one another as the cell continues to grow. Eventually, those DNA molecules end up on opposite sides of the cell. Division of the cytoplasm and synthesis of new cell wall occurs here at the midline, and we end up with two daughter cells, both of which are identical copies of the parent cell that we originally started with. Binary fission is primarily used by prokaryotic organisms, but we should note that it is also used by some protists. We're not going to look at them. They do weird things with their genomes and are an exception to the rule. But in general, binary fission is limited to those very structurally simple organisms in the prokaryotic domains. Now, once we move on to more complex eukaryotic cells that have that extensive compartmentalization and organelle structure, as well as a more complicated-looking genome, binary fission is no longer sufficient to serve those cells. Remember, the eukaryotic cells would be the fungi, protists, plants, and animals. The cell needs to accurately and with high integrity segregate the cellular contents into two daughter cells, and so a more complicated mode of cell division evolved, and that's mitosis. Mitosis is a complex cell division process that's used by eukaryotic organisms to ensure that their multiple, duplicated, linear chromosomes and all of their cytoplasmic organelles get appropriately segregated during the process of cell division. Mitosis occurs in single-celled eukaryotes like yeast, and like we saw in prokaryotic organisms with binary fission, The purpose of that mitotic event is going to be propagation of the species. When a yeast cell divides by mitosis, it produces 2 new daughter cells and so essentially continues on that population. And just like we saw with binary fission, mitosis is going to be asexual reproduction, so the daughter cells are actually clones of the original parent cell. Like we saw with our prokaryotes undergoing binary fission, the rate of mitosis in this example is rate limited primarily by environmental conditions, so as long as nutrients are plentiful and wastes aren't in excess, then single-celled eukaryotes like yeast will be able to continue to divide through mitosis. But we also see mitosis being used in multicellular eukaryotes like plants and animals for cell division as well. But in those cases, the purpose of mitosis is not to create new organisms, because most of that is through sexual reproduction, and a specialized population of reproductive cells is required for that. But instead, mitosis is being used to expand out the cell population. So for example, during early embryogenesis, we start with one fertilized egg, and in the adult organism, we end up with tens of trillions of cells. This expansion in the cell population occurs through mitosis. It's also used to replace cells that have become damaged or worn out or old, or that are lost through injury. The daughter cells are going to be clones of the parent cell, so clonal expansion of the cell population during growth and development and replacing worn-out cells are both types of functions that are associated with mitotic division in eukaryotic organisms. A third form of cell division, called meiosis, is used by sexually reproducing eukaryotes to generate their reproductive cells, or gametes. So if we're talking about animals, we'd be talking about sperm and egg. Those are produced in specialized reproductive organs, the ovary and the testes, from a specialized population of cells called germ cells. Germ cells would undergo a meiotic division rather than mitosis to produce gametes. And this can actually happen in both single-celled and multicellular eukaryotes. The important thing to recognize about meiosis is that this is a reductive division, and what's being reduced here is the amount of DNA. Before any cell division event happens, DNA has to be replicated because we're going to be creating daughter cells, each of which needs an instruction manual. In both binary fission and mitosis, the DNA is copied once and then divided up once. And so we end up back where we started. But in meiosis, the DNA is copied once, then the cells undergo 2 divisions that results in daughter cells that have half the genetic material of the original parent cell. A cell that has half the genetic material of the starting cell is referred to as a haploid cell. We're going to talk more about meiosis and the mechanism of meiosis in the next topic. But this is just sort of an introduction to meiosis and how it differs from mitosis. The other thing that we're going to see in meiosis is that genetic diversity is introduced in a couple of ways. In every meiotic division, we get new combinations of parental chromosomes that didn't previously exist, as well as new combinations of gene versions or alleles that are going to be introduced. So file that away. Genetic diversity is associated with meiosis, and identical clones are what we associate with mitosis. For the rest of this discussion, we're going to focus on mitosis. The first thing we want to understand is the context in which mitosis is occurring. And we have to understand that the vast majority of the life of a eukaryotic cell is not spent dividing. It's just spent carrying out normal cellular functions that are associated with that particular type of cell. If we think about the life cycle of a cell, what's commonly referred to as the eukaryotic cell cycle, the mitotic phase would represent that part of the cell cycle where the cell is actively undergoing division. But for the whole rest of the life cycle of the cell, what we would call interphase, the cell is not actively dividing. The cell maybe is just carrying out normal cellular functions, any specialized functions that are associated with that particular cell type, just sort of hanging out being a cell. Or it may actually be preparing for the next round of cell division. Interphase is actually divided up into 3 subphases that are called G1 for gap 1, S phase for synthesis, and G2 for gap 2. Let's imagine that we have a cell that's been produced by a cell division event. This cell is going to be entering interphase, and we're specifically entering into this first gap or G1 phase, where the cell continues to grow. It's going to be metabolically active, it's going to be expressing genes, it's going to be synthesizing proteins, it's going to be doing anything that is associated with this specific cell type. Only if that cell is signaled to do so is it actually going to leave G1 and enter the next phase of interphase, what's called S phase. At this point, the cell is actually preparing for the next round of cell division. And step one in preparing for the next round of cell division is to replicate or copy cellular DNA so that the daughter cells produced by that cell division event are both going to get a copy of that genetic blueprint. As the cell proceeds into S phase, the DNA would be copied, and from there the cell would enter into GAP2 or G2 phase, which is another growth phase where final preparations for cell division are going to occur. The cytoplasmic organelles are going to be duplicated, and importantly, any final repair to the DNA and checks on the integrity of the cellular DNA are going to happen here in G2 as well. Once the cell has double-checked that all cellular DNA has been correctly replicated and any mutations or damage that the DNA has incurred has been repaired, only then will the cell proceed into the mitotic phase. The mitotic phase is divided into two general subphases called mitosis and cytokinesis. Mitosis is the subphase in which the nuclear contents are being divided up, and cytokinesis is the part of the mitotic phase where the cytoplasm is being divided up and two new daughter cells are being produced. These two subphases are sequential. First, the nucleus is divided up, and then the cell splits in two. Mitosis itself has different subphases associated with it. The cell has to go through a sequential series of steps to make sure that the nuclear contents are appropriately segregated. Before we move on and actually start looking at the mitotic phase and seeing what those steps are, I do want to mention that for some cell populations, once they've been produced and they've become specialized, they undergo what's referred to as an exit from the cell cycle. So that's sort of represented by this arrow here. And they enter into what's sometimes referred to as G0, but what's really just G1 forever. These cells are referred to as being terminally differentiated. A good example of cells that become terminally differentiated and don't divide anymore are neurons. As we've talked about before, the extensive connectivity between neurons is really important for the communication and control that's associated with the nervous system. So as you can imagine, if you've got a neuron that's got hundreds of connections with other neurons, if it were to undergo cell division, it would actually have to lose all of those connections. To divide and produce two new daughter cells and then try to reestablish all of those connections, that would massively disrupt the function of the nervous system. So once neurons differentiate, those cells exit the cell cycle and go into G0. Before we start talking about all of the events that go on during mitosis, and I should say that we're going to spend most of our discussion on this and not on cytokinesis, frankly because not as much is known about how the division of the cytoplasm and the cytoplasmic organelles is regulated. We need to remind ourselves what DNA looks like and how it actually exists in a eukaryotic cell, because that becomes very germane to this discussion. First of all, let's just make sure that we understand what's happening with the DNA as it proceeds through the cell cycle. So let's start with this cell right here that's just been produced from a mitotic event. Two new daughter cells were produced, but we're just going to follow what happens with this one. So let's pretend that this cell actually has two DNA molecules in its nucleus. Those are represented by these straight lines. Obviously, most cells have a whole lot more than two DNA molecules, but this is just to illustrate what happens. The DNA contains the cellular genes that are being expressed and copied and made into mRNA. And that mRNA is coming out here into the cytoplasm, being read by ribosomes and translated to build a protein, as we've talked about many times before. The cell is just hanging out being a cell, but if the cell receives a signal, it's going to proceed into the next phase, S phase, and it's going to start preparing for the next round of cell division and begin copying its DNA. We're going to look at the details of DNA replication in a whole lot more detail later on, but for right now, we just want to understand that each of these DNA molecules in this cell are going to be copied, and what we're going to have then is our DNA held within the nucleus, but instead of one DNA molecule, we're going to have two identical copies of DNA. Each of these DNA molecules is going to be copied, and we now have two identical copies. These two identical copies are referred to as sister chromatids. These sister chromatids are held together by encircling proteins called cohesins. these little hoop-shaped proteins. And they're not allowed to separate until we get to the mitotic phase. So all the way through G2, those two copies are going to be held together and not allowed to separate from one another. In this way, the cell is actually keeping the copies together until it's ready for them to be pulled apart. The cohesins hold the copies, chromatids, together throughout G2 while the cell makes final preparations for division and repairs any mistakes in the DNA prior to division. Remember that in eukaryotes, so-called naked DNA is not present in the cell. Instead, what we find is that the DNA is compacted and condensed by its interaction with histone proteins into a form called chromatin, the complex of DNA wrapped around proteins coiled up and condensed and organized within the eukaryotic nucleus. That's what's being represented here. In blue is the strand of DNA, and here we can see the successive levels of increased condensation and compaction that can be associated with the structure of chromatin in the cell. So these little purple squiggly guys are meant to represent the histone proteins that the DNA gets wrapped around. Here's a more accurate red structural representation of what that looks like. The DNA is the double helix here in the gray, and then these colorful alpha helices here are representing the protein structures of the histone proteins making up the core of that nucleosome. Notice that each of those histone proteins actually has a little squiggly N-terminal tail that projects out from the nucleosome, and that's represented here by these little tails that stick out. As we're going to see later on in more detail, modification of those histone tails is an important means of regulating gene expression. The first level of chromatin compaction and organization is just wrapping the DNA twice around a core of histone proteins. Then that actually gets coiled up into the cylindrical structure, sometimes called the selenoid structure. So we go from a 2 nanometer in diameter DNA molecule to a 10 nanometer in diameter nucleosome, and then a 30 nanometer in diameter fiber. These 30 nanometer fibers are then looped and coiled up around a central protein scaffold to form an even larger fiber. This represents a higher level ordering or condensation or compaction of the DNA, and then these 300 nanometer fibers can then get sort of coiled up and compacted even further. But this level of compaction that we see from the 30 nanometer fiber up to this very, very tightly coiled and compacted form of chromatin, we really only see in the context of cell division. In a normal cell that's in interphase and not dividing, this would be the structure of the DNA. It would be this 30 nanometer fiber form. And this makes it easy to unwrap and express those genes in that region of the DNA if that particular protein that gene encodes needs to be expressed in the cell. What we see happening at that transition from interphase into the mitotic phase as the cell is entering mitosis is a dramatic shift in the structure of the chromatin, from this 30 nanometer fiber structure of chromatin to this very, very tightly coiled and compacted form of chromatin that we really only ever see in the context of cellular division. We see the DNA change in its structure from a state where we really can't make out any individual DNA molecules, like we see here in the upper right cell. So this would be a cell that's in interphase. We don't really see any distinct DNA molecules. Whereas here in this cell that's entering mitotic division, we can make out these distinct little fat fingers of DNA, and that again represents this very highly coiled and condensed state. These two sister chromatids are now highly compacted and coiled up and condensed, still held very tightly with one another by those cohesin proteins until they're allowed to separate. Those circular cohesins would actually be all along the DNA holding these sister chromatids together, but they're particularly concentrated here at a region of the chromosome that is called the centromere. The centromere is a specialized region of DNA where microtubules are actually going to be attaching and driving the separation of these duplicated DNA molecules from one another. We see a particularly high concentration of those cohesin proteins encircling the DNA here. And as the cell proceeds through the mitotic division, the cohesins that are holding the arms of the chromosomes together can actually break and come loose. But in this region at the centromere, the two sister chromatids are going to be held very tightly to one another until the cell gets to mitotic division and those DNA molecules are allowed to separate from one another completely. Here's another visualization of the structural change that we see as a cell enters the mitotic phase. So here's a set of chromosomes as they might appear in interphase. These are G1 chromosomes. And then as the cell enters into the mitotic phase, we go from this very extended, stringy, and decondensed state of chromatin to this very tightly coiled and compacted and condensed state of chromatin. Here's a view of what those cohesins actually look like and how they're encircling the DNA molecules. Just to get your sense of scale here, remember that I said that very tightly coiled chromatin structure is actually loops of these 30 nanometer fibers arranged around a central core. So this would be two sister chromatids held very tightly next to one another, and these would be their individual loops of chromatin. Blue would be one chromatid, purple would be the other one, and some of their loops are actually interwoven with one another, what's referred to as concatenation. It's these loops that would be encircled by those cohesin proteins to hold these two highly compacted and condensed DNA molecules together. So it's not that we have these humongous protein complexes that are encircling the whole giant 1400 nanometer wide chromosome itself. It's that we have these smaller proteins that are sort of at this region where the two sister chromatids are interwoven with one another. Now let's see what happens as a cell proceeds to divide up its DNA. This is a fluorescence micrograph, a light microscopy photo of a living cell in which two types of proteins have been labeled with fluorescence. The first protein here in orange is the histone proteins. And so those are the ones that are associated with the DNA molecules. Then in green, we have fluorescent labeling of the microtubule network in the cytoplasm of the cell. As we talked about before, microtubules have an essential function to play in the cell, in that they form what are essentially superhighways within the cell that motor proteins can transport materials along. During mitotic division and meiotic division, the microtubule network serves as the transport track for the DNA molecules that are being segregated and sent to two different daughter cells. And what we see as the cell enters into mitosis is a massive reorganization of that microtubule network. The green fluorescent labeling is going to allow us to visualize that reorganization. Here we're starting with a cell in interphase. It's not actively dividing. You can see that its microtubule network is sort of in this loosely interconnected state throughout the cytoplasm. But then as the cell gets that signal to start preparing for division, it's going to copy cellular DNA in S phase, and once it's gone through G2 and verified the integrity of the DNA and verified that the cell has grown enough and that the cytoplasmic organelles have all been duplicated, it's going to proceed into mitosis. The first subphase of mitosis is referred to as prophase. And here, the DNA starts to condense and go into that very, very tightly coiled and compacted state that we were just looking at. Then the cell enters into the next subphase of mitosis, what's called prometaphase. In prometaphase, a couple of things happen. We start to see the microtubule network being extensively reorganized from that network-like structure into a structure that's referred to as the spindle apparatus that you can get a better look at in the next phase here. That reorganization is controlled and organized by this circular structure you can see here, and that is the centrosome, which is a type of microtubule organizing center found in animal cells. There are different types of microtubule organizing centers in other types of cells. In animal cells, it just so happens to be a centrosome. If we zoomed in, what we would see inside the centrosome is two bundles of microtubules called centrioles, and that's where each of these microtubule fibers is actually originating from. The cell has two centrosomes that are here very close to the nuclear envelope. And in prometaphase, what we start to see is that these centrosomes start moving apart from one another so that they end up on opposite sides of this nucleus, and they reorganize the microtubules so that they're sending out this radial array of microtubules from the center of the centrosome. As the nuclear envelope starts to fragment and that double phospholipid bilayer structure starts to be disintegrated, these microtubules start reaching in and forming attachments to the DNA molecules that were present in the nucleus but that are now exposed. Let's zoom in and take a closer look at what happens during prometaphase when these microtubules start reaching out and making their attachments to those sister chromatids in preparation for separating them and sending them to two daughter cells. This diagram is meant to represent one chromosome that is comprised of two sister chromatids. These squiggly bits here would be the sister chromatids, and these ring-shaped proteins here in the middle would be our cohesin proteins that we've talked about before. As I said before, the highest concentration of those cohesin proteins is going to be in the region of the DNA referred to as the centromere. The centromere isn't only characterized by the fact that it has a large concentration of cohesin proteins. It's also the site where we see assembly of a protein complex called the kinetochore. Here in red, I'm just drawing a blobby set of proteins binding and assembling on the DNA in this region of the DNA called the centromere. What's going to happen is that each centrosome is going to send out microtubules, and there will be about a dozen or so microtubules that reach out and connect up with this complex called the kinetochore on the centromere of each sister chromatid. I'm only drawing a few lines here, but there would be about a dozen or so microtubules connecting up to each kinetochore. And if you recall from earlier in the semester, you'll remember that the way that transport happens down these microtubules is through the activity of motor proteins. So we have to imagine these little motor proteins that are walking along these microtubules, and they're going to be connecting up with these kinetochore proteins. And when the cell is ready for these two sister chromatids to be separated from one another, these motor proteins are going to be pulling in opposite directions towards the opposite poles of the cell from which the microtubules are radiating. The motor proteins are trying to pull these chromatids apart from one another, but they can't separate them. Why? Because those cohesins are still intact and are preventing these chromatids from being separated from one another. So essentially what you get is this tug of war where these motor proteins are pulling this way, these motor proteins are pulling that way, and nobody's really making any progress, and that generates force on the chromosomes. When everybody's attached up to the kinetochore, and motor proteins are assembled on either side and pulling equally in opposite directions, then we end up at a stage called metaphase, where the chromosomes here in orange are actually aligned at the midline of the cell. What we can't see here are the motor proteins that are trying to tug them apart, but they're sort of at a standoff because the cohesins are preventing the separation of these DNA molecules. Once the cell has detected that equal tension is present on each of the sister chromatids, that is, that they are correctly connected up at their kinetochore to the microtubule network, then the cohesins will be cleaved, and as soon as that happens, there's nothing preventing these motor proteins from being able to pull. So that's exactly what's going to happen. These motor proteins are going to start pulling in opposite directions, and we're going to see the sister chromatids being pulled apart and the DNA segregated and pulled to opposite poles of the cell. At this point, we say that the cell is in anaphase. Following anaphase, the nuclear envelope is going to start to reform around this set of DNA at each end of the cell. And then ultimately, what we're going to see is cytokinesis, where the cell membrane gets pinched in an even tighter circle between these two ends of the cell, effectively pinching off the cell to form two new daughter cells. Here's an electron micrograph showing a cell undergoing cytokinesis. And what we can see is that the plasma membrane is actually being pulled in, forming this furrow referred to as the cleavage furrow. And what's happening is very similar to what happens in muscle cell contraction. We have microfilaments, in this case a microfilament ring, that gets pulled on by motor proteins in such a way that the ring of microfilaments get closer together. The ring of microfilaments is attached to the plasma membrane, and as that ring gets smaller and smaller and smaller, it pulls the plasma membrane in, and we effectively see a pinching off of the plasma membrane, resulting in separation of the cytoplasm and two new daughter cells. Obviously, there has to be a very, very tightly controlled set of steps ensuring that the Golgi apparatus is duplicated and segregated appropriately to these two daughter cells, and that the smooth ER and rough ER are segregated appropriately as well. We're not going to go into any of those details because very little is known about how that's accomplished. We know a lot about how the DNA is divided up because that's something you can observe very easily just using a light microscope, so people have been looking at that for a really long time. But the technological tools to investigate how Golgi separation and separation of those other cellular organelles is happening have only recently started to become available, and so that's a very active area of research right now. Now we'll see a short video of some cells undergoing mitosis so that we can see what this looks like in real time. As you'll see, it's not actually a set of discrete steps. It's one continuous process that, once it begins, sort of flows from beginning to end in a very dynamic fashion. What we see here are two kidney epithelial cells. And this cell up at the top is going to stay in interphase. It's not going to be dividing. But this cell down here at the bottom is getting ready to undergo mitosis. We're going to view this a couple of times, and I want you to pay attention to what's happening here in the nucleus. Again, the histone proteins that are complexed with the DNA in chromatin are labeled with fluorescent orange, and the microtubules are labeled with fluorescent green. As this cell enters mitosis, the highly compacted form of DNA is starting to form. Those chromosomes that are highly compacted become visible, and you're also going to see that the cellular microtubule network gets massively reorganized by these centrosomes that are present right here at the nuclear envelope. Let's watch this a couple of times, and I want you to pay attention to what happens with the DNA because we're going to see something very interesting. Okay, let's watch that again, and I'm going to pause it at key points. At this point, we've seen the chromosomes condense, and you've seen the microtubules get reorganized. And what we're seeing at this stage is that the microtubules are starting to form their attachments to the kinetochores and starting to tug on these chromosomes, and that's why they're wiggling around. You can see pretty early on that most of the chromosomes get connected up correctly, so they're being tugged from opposite directions. But look at this guy down here. You see what's happening with this DNA molecule? It's not connected up correctly. It's being pulled only from this centrosome, but not from this centrosome. And so the cell actually has to wait for that DNA molecule to get connected up correctly before it proceeds to anaphase and telophase in the rest of cell division. We'll watch it one more time. So tugging, microtubules connecting up to kinetochores, and motor proteins pulling, and everybody's getting connected up and pulled to the midline of the cell, but the cell doesn't divide yet because that chromosome was not connected. As soon as it gets connected up and tugged to the middle of the cell gets the signal that equal tension is present and all chromosomes are correctly connected, and the cell proceeds to anaphase. As you can see, the process of mitosis is very dynamic, and once it begins, it sort of flows from beginning to end in a very smooth and dynamic fashion. As soon as certain molecular events have been accomplished, then the cell is ready to move on to the next stage, and to the next, and to the next, and ultimately to cytokinesis, resulting in the separation of those newly produced cells into distinct cells. Even though it's really useful to divide up mitosis into these subphases, prophase, prometaphase, metaphase, anaphase, telophase, etc., in some sense they're kind of arbitrary distinctions and boundaries. They're ways to describe the events that are happening during mitosis in a coherent fashion, but it really flows from beginning to end. We've just seen mitosis as it would happen in an animal cell. How does this differ from mitosis in a plant cell? As it turns out, the basic mechanism of mitosis is exactly the same as what we would see in an animal cell. The subphases of mitosis would be identical. So we would start with a cell in interphase. So this is a plant cell just hanging out being a plant cell. If it gets a signal that it needs to divide to produce more cells in this localized region, we're going to see duplication of the DNA in S phase, exactly the same way that we talked about. We're going to see double checking that DNA replication and repair are complete in G2, and then proceed into the mitotic phase. The first subphases of the mitotic phase are going to be prophase and prometaphase. I'm just going to call that prophase here, where we would see condensation of the chromosomes, fragmentation of the nuclear envelope, and reorganization of the microtubules into a spindle-like structure. Here's one of the differences that we would see in plant cells. You can't really see it in the microscopy image here, but plant cells don't have centrosomes. They organize their microtubule network in a different fashion. It actually relies on a ring of microfilaments that are here in the midline of the cell, and so organization of the microtubule network is a little bit different, but it does get reorganized, and motor proteins are still going to attach up to the chromosomes. We're still going to see that tug of war, and eventually we're going to see alignment of the chromosomes at the midline of the cell. Once the cell has received the signal that equal tension has been accomplished, everybody's lined up in the middle, those cohesin proteins are going to get cleaved, and we're going to proceed to anaphase, resulting in the sister chromatids being pulled to opposite poles of the cell. Then telophase, in which new nuclear envelopes are reformed around these two sets of segregated DNA. And eventually we see cytokinesis, which divides this one cell into two daughter cells, each with its own nucleus and its own set of genetic instructions. Now here's a second difference that we see coming into play here at telophase and cytokinesis, sort of combined here at the end. You'll recall that plant cells have a cell wall made up largely of cellulose. So in cytokinesis, it's not enough to just pinch off the plasma membrane. These two daughter cells have to have new cell wall built between them. The way that's accomplished is that the plant cell will lay down this little row of vesicles, and inside the vesicles is cellulose and the other components of the plant cell wall. Once this line of vesicles is formed in the middle, and then those vesicles fuse with one another, what we end up with is plasma membrane extending from one side of the cell to the other, and in the middle, what we're illustrating in orange here, would be what was inside the vesicles, which is the cellulose and other cell wall components. This is referred to as the cell plate, and you can actually see that very nicely in microscopy images using a stain that will identify cell wall components, cellulose and other molecules. So those are the main differences that we would see in plant cells versus animal cells. If you're interested, I've given you a link in the PowerPoint slides to a very nice review article that talks about how the plant spindle is formed. And it gives you some details of the molecular mechanisms that allow plant spindle formation in the absence of centrosomes, which is something that's really only been understood in the last 10 years or so. In this video, I've given an introduction to 3 different mechanisms of cell division that are used in different contexts and in different organisms. We've seen how binary fission works, we've taken a look at the molecular events that occur during mitosis, and we've compared mitosis in plant and animal cells. In the next topic, we'll talk about regulation of the progression of a cell through the cell cycle, so that cells only divide when they're told to, and behave like good citizens in the context of the organism as a whole.